In this video, we will cover how to carry out the pre-processing that mode requires for visualization. In the first drop-down, select which column contains your molecular identifiers. Next, if a sample information file was not uploaded, assign groups to samples by switching the toggle from No to Yes. Here, in the text box, enter the names of your groups, separated by a comma in order. For example, if you had an experiment with a control and two infection conditions, you may type control, infection A, and infection B. Make sure the number of entries matches the number of columns in your expression data file, minus the one column that contains the molecular identifiers. Click Confirm when ready. Next, a table will appear under sample data with the sample names and the group designation. If you would like to remove a sample, leave the group set to NA. We recognize that for 20 or more samples, this may feel cumbersome, and recommend users build a sample information file instead. The next few options are only applicable if the expression data are abundances from mass spectrometry or nuclear magnetic resonance, as opposed to RNA-seq transcript counts. Here, denote what value represents missing data in your dataset, which is typically NA. Set the data scale. Currently, raw intensity, log base 2, log base 10, and natural log are supported. If the original data scale is raw intensity, select either log base 2, log base 10, or natural log. Data will then be log transformed by this specification. Next, regardless of data type, if a differential statistics file was uploaded, select which columns denote the full change and p-value columns. Note that for abundance data only, there can be two p-value columns, one for the ANOVA and the other for the g-test. Make sure the names of your comparisons are consistent. If this step is done correctly, there will be a comparison table below the statistics table in the Preview Tables page. Here is an example of that comparison table for MS or NMR data that is generated in Mode. The first column denotes the detected comparison, the second column indicates the corresponding fold change column, and the third and fourth columns indicate the corresponding p-value columns for the ANOVA and the g-test respectively. If this table does not appear, you have not formatted the differential expression data correctly. Clicking Check Input should help you determine what needs to be fixed about these column names. If still unsure, see our video titled Understanding Statistics Results, which has a link in the video description. Once done, click Check Inputs. If the next step in the sidebar opens, called Normalize Data, you have properly conducted this step.